Hey, welcome, Miss Ray here. So we're here today. We're going to be talking about the cell cycle, which is the cycle of growth and division of cells in living things. The notes for this start on page 27 of your blue book. So if you have a book you want to follow along, this starts on page 27. Alright, so to begin, just some fun facts. All humans are roughly 99.9% identical, which means only 0.1% of our genetic makeup differs from person to person. As far as number of chromosomes, humans have 46 chromosomes, goldfish have 94, dogs have 78 chromosomes, and some ferns, those are green plants, can have up to 1,020. What this tells us is that the number of chromosomes does not determine the complexity of living things. Humans are much more complex than goldfish, dogs, or ferns, but all goldfish, dogs, and ferns do have more chromosomes than humans have. So complexity and chromosome number are not related. Identical twins do not have identical fingerprints. Humans do share some genes with bananas. And if all of the DNA in our whole body were laid out in a straight line, it would stretch to the sun and back over 600 times. You have to think about all the DNA in every cell, and we have millions of cells, which is a lot. So our first question is true or false? The more chromosomes an organism has, the more complex it is. So that's question one. So just to give you some context, every person, every living thing, no matter how large or how small, started out as a cell, smaller than the period at the end of a sentence. So smaller than this. We all started out as one cell, smaller than that period. But over time, that one cell divided into two. The 2 divided into 4, the 4 divided into 8, and so on and so forth until we came to have as many cells as we have today. So there's many reasons that cells divide. First off, we make more cells for having more cells in our bodies. It's a type of asexual reproduction, making identical copies of our cells to help us grow. Unicellular organisms do this by making identical clones. In humans, we need to actually grow from one cell to many cells, or from the size that we see as when we're a baby to the adult human. And this happens from a fertilized egg into the multicellular organisms that we see today. We also need to make new cells anytime that we have to repair something in our bodies. So that could be to replace cells that just die from normal causes. Our cells don't live but so long, they're not immortal. So eventually all of our cells will die, and we have to replace those cells when they die. But it also could come from wear and tear or from an injury. So if we were to fall and scrape our knee, we would have to repair those cells. Kind of like when you break a bone. Your bone cells have to repair to fix the bone. Some organisms, like lizards, can actually replace whole appendages or tails. Humans can't do that. We can't replace any limbs or the appendages that fall off, but we can repair ourselves when we are injured. So question number 2, why? Select all that apply. Why do cells need to divide? So why do cells need to divide? So in order for a cell to actually divide, several things have to be copied. We can't just take our one cell and split it into 2. There wouldn't be enough to make 2 cells out of it. So before a cell can divide, we have to make copies of DNA, copies of organelles, copies of the cell membrane, and copies of enzymes so that both cells have what they need in the end. So we're going to take a peek at our chromosomes. We talked about chromosomes last time. And we have to duplicate our DNA. We have to duplicate our chromosomes. And our duplicated chromosomes consist of 2 sister chromatids. They each contain identical copies. So in this image, we see our chromosome at the top, So this is one chromosome by itself. This is what a chromosome looks like. When a chromosome is duplicated, it now has two identical sister chromatids. So this is a duplicated chromosome. When we have the two chromatids, that is a duplicated chromosome. After our cells divide, one chromatid goes into each new cell, and we're back to just that one chromosome that we started with. So we start with one chromosome, we make a copy of it to prepare for division. When it's copied, it has two sister chromatids. Then we'll separate these two, one for each cell. And then we're going to come back to this organelle we haven't talked about in a really long time, called centrioles. Centrioles are very, very important in division. When we are making our new cells, we have to be very, very careful when we split our DNA, when we split our chromosomes, to make sure that our two cells come out absolutely identical. So centrioles, along with some spindle fibers, are going to help guide chromosomes during each stage of cell division. So centrioles make spindle fibers, and those fibers are going to help move chromosomes during cell division. This is to make sure that we have identical cells in the end. Question 3. What do centrioles do during cell division? So let's just do a quick overview of the cell cycle. So this is what our cell cycle will look like. Here we have our normal cell doing its everyday thing. That stage is called interphase. Interphase, our cell doing our normal thing. Then our cell starts to do something interesting when it's ready to divide. We see our spindle fibers start to release. These are our centrioles. We now start to see our chromosomes, and the nucleus starts to disappear. This is called prophase. Next, all of our duplicated chromosomes are going to line up along the middle. That is called metaphase, M for middle. Then we're going to start to pull our duplicated chromosomes apart, again, because only one of each goes in each cell. So our duplicated chromosomes split apart. When they split apart, that is called anaphase. And then our cell is going to start to split into two, where our nucleus comes back. Our cell starts to pinch, and that is called telophase. We're going to talk about each of these in a second. And then our cell is actually going to complete splitting. It's going to split into half, and when it completely splits in half, that's called cytokinesis. And again, we're going to talk through these. So step 1 was interphase. Interphase, it's 90% of the cell cycle. A cell spends 90% of its time in interphase. And interphase is just a cell doing its normal thing. So a cell isn't dividing all the time. Most of the time, your cell is just hanging out, doing its everyday job. In this process, it's making RNA, it's making proteins, making enzymes. But it does have to start making some preparations if it needs to divide. So when the cell needs to divide, it will start to do a few things. So when it's ready to divide during interphase, there are 3 different phases that can happen. G1 is growth phase 1. So your cell will do its everyday job. It's doing what it usually would do. So growth phase, gap phase 1 is cell doing what it would typically do. The cell might also grow. Growing a little bit just to prepare to divide. Then it has to copy the DNA. DNA synthesis, or S phase, S for synthesis. DNA synthesis is when the DNA is copied. So all of your DNA is copied. And then in growth phase 2, your cell is preparing for division. It's making sure it did everything properly before it actually divides. This is where you're going to also copy organelles. So the cell is going to grow a little bit more. You're going to make extra membranes so that you're perfectly ready to start division when the time comes. So this is interphase. Growth phase 1, G1, cell doing its normal everyday thing, cell grows a little bit. Then it will move into S phase, where DNA is copied. After all of the DNA is copied, it will move into G2, growth phase 2. The cell grows some more, makes organelles, proteins, membranes, make sure that it's perfectly ready to start division. This all is considered interphase in that 90% of the cell cycle. So what is a cell going to look like during interphase? So here's our cell during interphase. It has a very well-defined nucleus. The nucleus is very large, very central. You can clearly see it. But the DNA is still loose, meaning it's kind of all balled up. We call that chromatin. We can't see the chromosomes yet. You start to see your spindles appear, so you see your centrioles with just a little bit of a spindle fiber starting to appear. So, question number 4. Select all that are true about interphase. So, select all that are true about interphase. So again, right before the cell is able to divide, our loose DNA has to be condensed or packaged into chromosomes. So our DNA will go from the double helix that we know into nice organized structures, which look like chromosomes. So chromosomes are essentially DNA that's wound very, very tightly together to make it easier to see. To smaller, easy, organized, packaged DNA is called a chromosome. So after interphase, if the cell needs to divide, then it's going to start mitosis. Mitosis is the division of the nucleus in a cell. It's the division of the nucleus because prior to mitosis, you have one cell. At the end of mitosis, you have one cell with 2 nuclei. So it's only the division of the nucleus. And mitosis is split into 4 phases. Those phases are prophase, metaphase, anaphase, and telophase. And we abbreviate those as PMAT. So again, mitosis, division of the cell nucleus, 4 phases, prophase, metaphase, anaphase, telophase, PMAT. Question 5. Which is mitosis? So what is mitosis? So let's look at these phases. We're going to start with prophase. During prophase, our DNA condenses. This is the first time you actually see chromosomes. We see our chromosomes during prophase. Chromosomes are visible. We're also going to see our centrioles start to move to opposite sides of the cell. The spindles are forming. We see our spindle fibers starting to stretch out. They're ready to do their work. The nucleolus disappears, which is that dark spot inside the nucleus, and the nuclear membrane starts to break down. So our nucleus is now dotted because it's starting to disappear. It's going to disappear so we can actually have access to this DNA to help move it around. So after prophase, our cell is going to go into metaphase. In metaphase, our nucleus is completely gone. No nucleus, the DNA is now in the cell in chromosomes. Remember, our DNA has already been duplicated, so each of these that we see is a duplicated chromosome. Half will go into one cell, half will go into the other in the end. During metaphase, the chromosomes line up in the middle, M for middle. Metaphase, M for middle. They line up in the middle because they line up in the middle so that when the cell splits, it's nice and organized. Half goes to one side, half goes to the other, and we'll see what that looks like. So again, meta means middle. This is going to help ensure that we separate properly. We only want one of each duplicated chromosome in the other cell. If they don't line up, they're just going to do stuff randomly. After metaphase, we move into anaphase, and this is where our duplicated chromosomes split apart. So we see our duplicated chromosomes being broken in half. So one chromatid of each duplicated chromosome will go into each new cell. The spindle fibers here are actually pulling them apart. They're separating those duplicated chromosomes, and now they're becoming individual chromosomes. The cell's going to start to elongate, and it looks like an egg. It's getting prepared to start to divide. It's not dividing yet, but it's getting prepared. It's elongating, getting ready for it. Our chromosomes are now moving to opposite poles of the cell. And to remember this, A for apart. A for apart, our chromosomes are moving apart. If our chromosomes do not properly separate, we're going to have mutations. This could lead to organisms with 23 chromosomes, kind of like we talked about when we talked about Down syndrome in our last in our last lecture. So this could lead to people having too many or too few chromosomes. Our last stage is telophase. Telophase is when our cell starts to look normal again and it's starting to pinch. So in telophase, chromosomes are at the opposite ends of the cell. Our nucleus is starting to reform. So we see our nucleus starting to come back in on both sides. Chromosomes are starting to unwind. We no longer see those visible chromosome X structures. The spindle fibers are now disappearing. Things are starting to look more normal. But we're also seeing the beginning parts of division where our cell is going to start to pinch a little bit. And again, this is the last step. Mitosis is done after telophase. We only have one cell, but two nuclei. So again, mitosis is nuclear division, the division of the nucleus. Question 6. During which phase of mitosis are the duplicated chromosomes pulled apart? Question 7. During which phase of mitosis do chromosomes start to become visible? Question 8. During which phase of mitosis does the nucleus start to reform? After mitosis is over, cytokinesis begins. Cytokinesis is the division of the cytoplasm. So this is when the cytoplasm physically splits into two. In animals, animal cells pinch. They pinch into two, and that pinching is called a cleavage furrow. They physically pinch into two. However, in plant cells, plants have a cell wall. They are unable to pinch. They can't pinch. So they form what is known as a cell plate. So essentially, a new cell wall forms along the cell plate in the middle to create two new cells. So after telophase, our cell will look like this. The cell plate will form a new cell wall right in between. So that will create two new cells. Question 9. What happens during cytokinesis? Question 10. Which type of cell will use a cell plate during division? So at the end of the whole cycle, we went through interphase, mitosis, and cytokinesis. We have new daughter cells. We get two exact identical copies of the original cell. They had the same DNA. They are clones. So we started with one cell, and we're going to end with two identical cells. Again, these are clones. This is a type of asexual reproduction. The cells are identical from the one in the beginning to the ones in the end. So a summary of the cell cycle. A cell spends 90% of its time in interphase. Interphase is split into three parts. G1 is when the cell will grow. S phase is when the DNA is replicated. That's called DNA synthesis. G2 is when it will grow a little bit more, copy organelles, copy membranes to prepare for division. That's interphase. After interphase, the cell will move into mitosis. Mitosis is when the nucleus divides. The process of dividing the nucleus happens in four parts: prophase, metaphase, anaphase, and telophase. Just after mitosis, the cell will physically divide during cytokinesis. This is where the cytoplasm divides into two, and we will now be ending with two identical cells. Just a reminder, our cells cannot live forever. Cells have their own lifespan. In human cells can only divide 40 to 60 times before they die. So our cells don't last forever. We need new ones to replace them, and that's one of the reasons why we need cell division. Our genes control the lifespan of the cell. The cell cycle also has several feedback checkpoints. These checkpoints are controlled by proteins, and their job is to ensure that cell division is happening properly. These checkpoints are going to make sure that there aren't errors. If there's an error in cell division, it could lead to cancer or mutations, and we don't want that. There are 3 checkpoints. At the end of G1, the cell is checked to see if it's capable of going into cell division. It will check again after the DNA has been copied. It checks again to make sure that the DNA has been copied correctly. And then at the end of mitosis, the cells will be checked again to ensure that there haven't been any errors. At any point, if any error is found, they can totally shut this down and kill the cell. They can just stop the division, and that can prevent lots of negative consequences. Question 11. What is the purpose of cell cycle checkpoints? There are some exceptions. Even though things are very organized and we have checkpoints, cells are capable of dividing continually. Sometimes things are missed, things aren't caught, and this can lead to cancer. And cancer is a disease that occurs when cells grow and divide at an abnormally high rate. They just continue growing and growing and growing, and then you have these lumps of cells, and those are tumors. So any type of damage to the DNA can trigger this type of growth. And it can happen anywhere in the body: the lungs, the breast, the skin, the stomach, the heart, colon, brain. It can happen anywhere in the body, anywhere that there's a specific mutation that causes continuous cell division. These are going to create tumors, which are just lumps of cells. The cell divides over and over and over and over and over, and eventually you have a large mass of cells, and that's called a tumor. But there are 2 types of tumors. The first type is called benign. A benign tumor is one that cannot spread. So it's just that one tumor. Benign tumors typically can be easily removed, well, depending on their location. Most benign tumors can be fairly easy to remove. Type number 2 is called malignant. Malignant tumors are tumors that can spread all over the body. And malignant tumors are harder to treat because oftentimes by the time you find one tumor, it's already spread somewhere else. And it can be very hard to treat because you have to track down all of those cells that could have spread to other places in the body. And that's when cancer becomes very hard to manage. Question 12. Which type of tumors are hard to manage because they spread easily? Question 13. What error in the cell can lead to cancer? And just to end, many things can cause cancer. Cancer can be genetic. There's some types that are more genetic than others, like breast cancer, colon cancer, and prostate cancer. Sometimes cancer can occur for really no reason that we can trigger. Sometimes it's caused by carcinogens or mutations. Some carcinogens are things that have been known to cause cancer, like smoking, asbestos, certain pesticides, mercury, and lead. Those can cause mutations, which can then lead to cancer. Radiation and X-rays can cause mutations that can also lead to cancer. So UV radiation, exposure to the sun, those can cause mutations in the skin that can lead to skin cancer. Remember your sunscreen. Also, certain types of viruses can also lead to cancer. Viruses that you might have heard of that can cause cancer are HPV and cervical cancer, as well as hepatitis. Another one that's not listed here is the AIDS virus. Those can also cause cancer. Please make sure to reach out to your teacher if you have any questions.